Welcome to another deep dive. Today we'll be focusing on a topic that I'm sure will be of great interest to all of our medical microbiologist listeners out there.
Definitely a topic close to my heart.
Yeah, so we're going to be dissecting Chapter 4 of Antibiotics Challenges Mechanisms, Opportunities by Walsh and Wensvitz.
Great book.
Yeah, and as you know, it really delves into the fascinating world of how antibiotics target Peptidoglycan assembly.
Yeah, one of the most vulnerable spots for bacteria.
Exactly. And for those of us on the front lines, a deep understanding of these mechanisms is absolutely crucial. So
It really is.
let's just jump right in.
Let's do it.
Walsh and Wensvitz kick things off with the beta-lactams, which, I always find fascinating. Even though penicillin is considered a classic, its mechanism is still so clever. Those suicide substrates are just brilliant.
Yeah, it's really elegant how those beta-lactams form those long-lived covalent bonds with the transpeptidases, essentially freezing the process of cell wall cross-linking for hours. It's incredibly effective, especially when you consider the speed at which normal cross-linking occurs.
It's like hitting the pause button on a high-speed assembly line.
Exactly.
Now, I know we're all familiar with the different generations of penicillins and cephalosporins.
Yes.
But I'd love to hear your perspective on why this continuous development was necessary.
Well, it all boils down to that constant arms race, As bacteria evolved to resist penicillin with enzymes like penicillinases, we had to respond. Methicillin, for instance, was a huge step forward in combating those penicillinase-producing bacteria.
Right.
But then, of course, new resistance mechanisms emerged.
Of course.
Leading to the development of ampicillin to broaden the spectrum of activity.
And then the cephalosporins came onto the scene, often boasting greater stability against those pesky beta-lactamases.
Exactly.
It's fascinating how this back and forth has driven so much innovation, leading to drugs like ceftaroline, which can tackle even the toughest opponents, like MRSA. But what I find even more interesting is how we've been able to revitalize some of those classic penicillins by combining them with beta-lactamase inhibitors.
Yeah.
Augmentin, for example, has given amoxicillin a new lease on life.
Absolutely, a classic example.
It really is a testament to the enduring power of understanding these fundamental mechanisms. Now, let's shift gears a bit. Walsh and Wensvitz point out this intriguing gap in our antibiotic arsenal.
Okay.
Transglycosylases, despite their crucial role in peptidoglycan synthesis, have been surprisingly difficult to target. It makes you wonder why.
It's a great question. I think the key lies in their large, extended active sites.
Okay.
These complex structures make it difficult to design small molecules that bind effectively and inhibit their function.
Yeah.
Monomycin, with its intricate pentasaccharide structure, is really the only natural product that has successfully cracked the code.
Monomycin is like a giant, intricate key fitting into a complex lock. And its effectiveness against gram-positive bacteria, especially S. aureus, really highlights the potential of targeting transglycosylases. However, as Walsh and Wensvitz discuss, its complexity also presents significant challenges.
Exactly. Its size and poor absorption make it a nightmare for clinical use. It's a shame because its nanomolar activity against these tough pathogens is truly impressive.
It's almost like having a powerful weapon that's too unwieldy to use effectively. But there's hope, right? Research into simplifying mitomycin, perhaps through total synthesis or biosynthetic pathway manipulation, could offer a way forward.
Absolutely. The fact that we have this natural blueprint in monomycin gives us a solid foundation to work from.
Okay, good.
Now, moving on to another fascinating target highlighted by Walsh and Wensvitz, let's talk about lipidicity. This molecule is like the Achilles heel of peptidoglycan synthesis.
It's incredible how such a central player has become vulnerable to such a diverse range of antibiotics.
Yeah, it's really a testament to the ingenuity of nature and our own scientific endeavors.
Yeah.
We have the glycopeptides like vancomycin, which, has been a lifesaver in treating serious gram-positive infections.
Yeah.
And then there are the antibiotics like nisin, each with their own unique way of latching onto lipid II and halting cell wall construction.
Vancomycin, with its complex heptapeptide structure, is a prime example. The way it forms that molecular cup to bind the D-L-terminus of lipid II is just fascinating.
Oh, it is amazing.
It's completely different from how nisin targets the Mernach phosphate region.
And the variety doesn't stop there. Ramiplanin, manopeptimycins, lysobactin. There's a whole arsenal of lipid II binders out there.
Wow.
Each with its own unique mechanism. It seems nature has found a multitude of ways to exploit this critical vulnerability. And what's even more interesting is that the bactoprenol cycle itself, which is responsible for transporting those peptidoglycan building blocks, offers additional points of attack.
It's like realizing that there are weak points throughout the entire supply chain. Walsh and Vensvitz even discuss familiar antibiotics like bacitracin, which, as you know, is often found in topical ointments.
Yes, but its mechanism is anything but simple. By sequestering bactoprenol PP bacitracin prevents the recycling of this crucial lipid carrier, effectively halting peptidoglycan synthesis.
It's like cutting off the fuel supply to a factory.
Exactly.
And then there's rhyolimycin B, another lipopeptide antibiotic,
Right.
but with a slightly different target, bactoprenol P.
Right.
The fact that we can target different intermediates in the cycle opens up a whole new avenue for antibiotic development.
It really highlights the importance of understanding these pathways holistically.
Yeah.
Now, for our final point in this section, let's delve into the fascinating world of peptidyl nucleosides, which directly target the memorrhea enzyme.
This is a class of antibiotics that's always piqued my interest.
Yeah.
Those complex structures like tunicamycin and muriatomycin are quite remarkable.
They're master impersonators.
Yeah.
These compounds mimic the UDP-Murnac pentapeptide substrate of memorrhea, effectively acting as competitive inhibitors. However, their complexity also presents a unique challenge.
That's right, the challenge of cell entry. Unlike the other antibiotics we've discussed, these molecules have to penetrate the bacterial membrane to reach their target.
Right.
Not an easy feat.
It's a hurdle that researchers are actively working to overcome. The potential of these compounds is enormous, but getting them where they need to be is a key focus of ongoing research.
It's a reminder that understanding the mechanisms of action is only half the battle. We also need to consider how these antibiotics interact with the bacterial cell and the challenges they face in reaching their targets. Now, speaking of challenges, bacteria aren't exactly passive in this fight. Let's move on to how bacteria respond to these attacks on their cell walls.
Right.
It's remarkable how they've evolved such sophisticated defense mechanisms. It's as if they've studied our playbook and developed countermeasures for each antibiotic strategy.
Yeah, it really does highlight the dynamic nature of this arms race, doesn't it?
It does.
It's not a static battlefield. Bacteria are constantly adapting, and we need to be equally agile in our response.
Absolutely. Walsh and Vensvitz bring up transcriptome analysis, which I find to be a game changer in understanding how bacteria mounts these defenses.
Honestly.
It's like being able to listen in on the bacteria's internal communications to see how they're reorganizing their forces in response to these attacks.
Exactly.
For example, studies on Streptomyces coelicolor, which ironically is a prolific antibiotic producer itself,
Right.
have revealed some fascinating insights.
Yeah.
So even the organisms that produce antibiotics have developed ways to defend against them.
Yeah.
That's incredible. What kind of responses have they observed?
When challenged with cell wall-targeting antibiotics like bacitracin, miantomycin, or vancomycin,
Right.
Streptomyces coelicolor activates a whole cascade of stress response pathways.
So it's like the cell goes into emergency mode,
Yeah,
mobilizing all its resources to survive.
exactly.
What are some of the key players in this bacterial defense system?
We see a significant upregulation of genes involved in cell envelope stress response,
Okay.
stringent response, heat shock response, and even oxidative stress response.
Oh.
These pathways are all interconnected, working together to protect the cell from the damage inflicted by these antibiotics.
It's fascinating how these seemingly disparate stress responses converge to combat a specific threat. It speaks to the interconnectedness of cellular processes.
Exactly. It's not just about patching up the cell wall. It's about a global shift in cellular priorities,
Right.
a reallocation of resources to ensure survival. And this understanding is crucial for developing more effective antibiotic strategies. We can't just focus on inhibiting a single target. We need to consider the broader cellular context and anticipate the bacteria's countermeasures.
It's like a game of chess, where we need to anticipate our opponent's moves and plan accordingly. This deeper understanding of bacterial response mechanisms is crucial in our quest to stay one step ahead.
Right.
Now, to really solidify our grasp of this complex world of antibiotic mechanisms and bacterial defenses, let's put our knowledge to the test.
Okay.
You ready for a little pop quiz?
I always enjoy a good challenge. Let's see what you've got.
All right, here are 5 exam-style questions for our astute medical microbiologist listener.
Okay, bring it on.
Question 1. Describe the mechanism of action of beta-lactam antibiotics. Using penicillin as an example, how does this differ from the action of vancomycin?
So thinking back to our discussion, how would you approach this question?
Well, I'd start by highlighting the fact that beta-lactams, like our good friend penicillin, target those crucial transpeptidases, also known as penicillin-binding proteins, or PBPs.
Right.
These are the enzymes responsible for the final cross-linking steps in peptidoglycan synthesis.
Precisely.
And the key to their effectiveness lies in their ability to mimic the natural substrate of these enzymes while harboring a hidden weapon, that reactive beta-lactam ring.
Absolutely.
This leads to the formation of a stable covalent bond with the active site of the transpeptidase effectively inactivating. It's like throwing a wrench into the gears of the bacterial cell wall assembly line.
Now, in contrast, vancomycin takes a different approach. Instead of targeting the enzyme directly, it focuses on the building blocks themselves, specifically lipid the second.
It's like intercepting the delivery truck before it reaches the construction site. Right. By binding to the D-ala terminus of lipid second, vancomycin prevents it from reaching the transpeptidases, effectively halting cell wall synthesis.
Two different strategies, both with the same devastating outcome for the bacteria.
Exactly. Now, for question 2, what makes moinomycin a unique antibiotic, and what challenges are associated with its development for clinical use?
Oh, that's a good one.
Moinomycin stands out because it's the only known natural product that effectively targets those elusive transglycosylases. Its large, complex structure is both its strength and its weakness.
That's an excellent point. It's able to bind the extensive active site of these enzymes.
Right.
But its size and complexity make it poorly absorbed and difficult to deliver effectively in the human body.
It's a classic case of a powerful weapon with logistical challenges.
It is.
Now, for question 3, why is lipid to second considered a key target for antibiotic development, and what are some examples of antibiotics that exploit this vulnerability?
Lipid 2 is like the linchpin in the entire peptidoglycan synthesis pathway. It's essential. It's exposed on the cell surface, and it has multiple binding sites that can be targeted.
It's a prime target for disruption.
It is.
We've got a whole array of antibiotics that have evolved to exploit this vulnerability.
We do.
The glycopeptides like vancomycin bind to the D-ala terminus,
Right.
while the lantibiotics like niacin target the Mernach phosphate region.
And the list goes on. Ramoplanin, manopeptimycins, lysobactin. There's a whole arsenal of lipid 2 binders,
It's amazing.
each with its own unique mechanism of action. It's a testament to the evolutionary pressure to target this critical molecule.
Now let's move on to question 4. Explain how bacitracin and phryolomycin B disrupt the bacterial cycle, highlighting their different targets.
This question dives into the crucial role of the bacterial cycle in peptidoglycan synthesis.
Right.
Bacitracin and phryolomycin B both target this cycle,
Yes.
but they have distinct mechanisms.
Right. Bacitracin, that common ingredient in topical ointments, actually targets bacterial PP, preventing its dephosphorylation and recycling back to the inner membrane.
It's like sabotaging the recycling plant.
Phryolomycin B, on the other hand, targets bacterial P, the form found on the outer face of the membrane, preventing it from being flipped back to the inner membrane.
Exactly.
So both disrupt the bacterial cycle, but by targeting different steps. And finally, question 5, what insights can be gained from analyzing bacterial transcriptome responses to antibiotics targeting peptidoglycan synthesis?
This is where we get to eavesdrop on the bacteria's internal conversations.
Right.
To see how they respond to these attacks on their cell walls. Transcriptome analysis allows us to see which genes are upregulated or downregulated in response to antibiotic stress.
It's like a window into their defense strategy.
It is.
We've learned that bacteria activate a whole cascade of stress response pathways,
Yes.
including the cell envelope stress response, the stringent response, heat shock response, and oxidative stress response.
This knowledge is crucial for developing more effective antibiotic therapies.
Right.
By understanding how bacteria adapt and evolve to overcome these challenges, we can design strategies to circumvent their defenses.
Absolutely.
Yeah.
It really is a remarkable dance between attack and counterattack, with both bacteria and scientists constantly pushing the boundaries of innovation.
And it underscores the importance of continued research and exploration in this field. You know, we can't afford to become complacent, especially with the rising threat of antibiotic resistance.
Absolutely. So to our dedicated medical microbiologist listener, I hope this deep dive into Chapter 4 of Walsh and Wensvich's Antibiotics, Challenges, Mechanisms, Opportunities, has been insightful and perhaps even sparked some new ideas.
It's a great book.
We've covered a lot of ground, from the classic mechanisms of beta-lactams to the intricate strategies of targeting lipid the second and the bacterial cycle. And of course, we delved into the fascinating world of bacterial responses, highlighting the importance of understanding their countermeasures.
Yeah, it's really crucial.
It's this constant interplay between understanding the mechanisms of antibiotics and anticipating bacterial resistance that will ultimately guide us toward developing more effective therapies.
Exactly. And for you on the front lines of patient care, this knowledge is paramount.
It is.
Keep asking those tough questions, keep exploring the latest research, and never underestimate the ingenuity of both bacteria and the scientists who are working tirelessly to combat them.
Remember, the world of antibiotics is constantly evolving, and it's up to all of us to stay informed and engaged in this crucial battle.
Until next time, stay curious.
It really is a remarkable dance between attack and counterattack, with both the bacteria and the scientists constantly pushing the boundaries of innovation.
And it underscores the importance of continued research and exploration in this field. We can't afford to become complacent, especially with the rising threat of antibiotic resistance.
Absolutely. So to our dedicated medical microbiologist listener, I hope this deep dive into Chapter 4 of Walsh and Wencevich's Antibiotics, Challenges, Mechanisms, Opportunities, has been insightful and perhaps even sparked some new ideas for you.
Yeah, a great book.
We've covered a lot of ground, from the classic mechanisms of beta-lactams to the intricate strategies of targeting lipid II and the back-to-prenol cycle. And of course, we delved into the fascinating world of bacterial responses, highlighting the importance of understanding their countermeasures.
Yeah, it's crucial.
It's this constant interplay between understanding the mechanisms of the antibiotics and anticipating bacterial resistance that will ultimately guide us toward developing more effective therapies.
Exactly. And for you on the front lines of patient care, this knowledge is paramount. Keep asking those tough questions, keep exploring the latest research, and never underestimate the ingenuity of both bacteria and the scientists who are working tirelessly to combat them.
Remember, the world of antibiotics is constantly evolving, and it's up to all of us to stay informed and engaged in this crucial battle.
Until next time, stay curious.
And thanks for joining us for another deep dive